Good evening, everyone. I'm Niketa. I'm a PhD student from IIT Madras, and I'm your TA for the course Basic Environmental Engineering and Pollution Abatement. So, this is week 7 of the live session. And before I start, I want to ask if you have any doubts from whatever lecture videos you watched for week 7. Okay, so if there are no doubts, then I'll proceed further. So, how the class goes like this. First, I'll give a brief intro and I'll explain some basic, I'll explain some selected topics. And once I'm done with that, we will be solving some questions and answers. So, in between, you're welcome to ask any questions or doubts if you have any. Okay, so moving on. So this is week 7, and so we've seen about wastewater treatment so far. So we've seen about primary, secondary, and tertiary treatment, and using the sequence. So now let's talk about activated sludge process. So can you explain what is activated sludge process? We have discussed this before. Okay, activated sludge process is a type of secondary treatment where the microbes are suspended and they are not stationary. So, what do we mean by activated sludge? So, basically, when we have a tank full of microbes degrading your organic waste, once the microbes reach a certain level of population, they start eating themselves because beyond that, they don't have any waste left and they enter a stage of self consumption. So they start dying, and when they die, they form sludge. So this sludge usually settles at the bottom of this tank. And when the wastewater is removed and it is transferred to the next process, we also remove the sludge and separate it. And this sludge will be again sent back to the tank. Why is it done? Any guesses? So I told you what is sludge. Now I'm asking, why is it recirculated?
Because of microorganisms presence.
Yeah. So the sludge contains a high concentration of living bacteria or living microorganisms as well. Because once the population exceeds, like along with the dead microbes, even live microbes will be present in the sludge. So these live microbes, they can repopulate the tank with when a fresh batch of wastewater enters it. So when that wastewater batch does not have enough microbes, then this sludge can be used to repopulate it. Okay, so this sludge, which contains activated microbes or activated bacteria, is your activated sludge. So activated sludge process means just that. You have some activated sludge which is collected at the bottom of the tank, and that is recirculated so that the bacteria will be coming inside. So the bacteria gets repopulated, and it is a suspended process. So we need a mixer. So mixer is usually provided, and then we also need some aeration so that the bacteria will survive. So we also provide air so that the bacteria can respire and it can continue to grow. Okay, so we have a equation. So we have an equation to represent this process, activated sludge process or ASP. So we can represent it in the form of an equation like this. I'll tell you what it is. So the rate at which the microbes are coming inside the system, or rate of inflow of microbes in the system, minus the rate of outflow of microbes from system, plus net rate of cell growth in tank is 0. Okay, so what this basically is, so this is your tank where activated sludge process is taking place. So we have an inlet, and through the inlet wastewater is coming in, and the process happens. So along with the wastewater there will be some bacteria present. So that is your bacteria which is coming in, or microbes coming in. Now, once they enter the tank, along with also we are adding some activated sludge also. So that sludge will also have some bacteria. So that is also entering. So Now these microbes, they will consume the organic matter and they will continue to grow and multiply. Okay, so if you have maybe 100 microbes entering the tank, once they consume the organic matter and continue to grow and multiply, maybe they will become 200 or 300 and so on. So they'll continue to grow and multiply. So uh, the and then once the process is done, there will be an outflow, and also there will be some sludge that is collected. So there will be microbes that are flowing out. Microbes will come out along with the treated water, and microbes will also come out along with the sludge. Okay, so how will you represent the total mass of bacteria at any time? So that can be represented through this equation. So the rate of inflow of microbes inside the system, or the rate at which microbes are entering the tank, minus the rate at which the microbes are going out. Okay, so that will leave us with the population which is growing inside. So if we if we subtract the rate at which the microbes are coming in minus the rate at which the microbes are going out plus the rate at which the microbes are growing in the tank, that will be equal to 0 at any time. Questions? Is it clear?
Yes, bro.
Okay. So this is because whatever microbes are coming in, they will grow, and along with the microbes that came in, and along with the some of the microbes that came in, and some of the along with that which are growing in the tank, if these two are exiting, then at any time there won't the concentration of the microbes inside the tank will be 0. And that's what this equation says. The rate at which the microbes are coming in minus the rate at which they are rate at which the microbes are coming in plus the rate at which they are growing inside. For example, if they are 100 and once they come in they become 300, and 300 microbes are going out in the form of sludge and along with the wastewater, then 300 minus 300 will be 0. So imagine this is 100, this is, we'll come to this later. So imagine the rate of cell growth in the tank. The extra organisms that grew in the tank is 200. So, and the rate at which the microbes are going out is 300. So totally it will be 0, right? That's what the equation says. So these are just approximate numbers. So it's actually a rate. So this can be further represented in the form of a formula. So Q into X naught minus Q by Q wasted into XE plus QW into XR plus RG slash V equals to 0. Okay. So this is the same equation. So here the concentration of microbes that are coming inside is X naught. And Q is the flow rate, and so that is this concentration multiplied by the flow rate will tell you how much mass of microbes that is entering. So this can be in terms of kilograms. So this term will tell you how many kilograms of microbes are entering into the tank. Now this minus now this term is the rate of outflow of microbes from the system. So this term is actually a sum of two expressions. So the first one is basically this is Q minus QW. So Q minus QW is Q is the flow rate of inlet, and QW is the flow rate of microbes to be wasted. So flow rate of microbes to be wasted is nothing but the outlet concentration. I mean, how much of microbes are going out or how much is exiting out of the tank. So that is QW. This multiplied by, so this term Q minus QW will tell you how much of flow is present inside the tank. So this is inlet, and this is the this is the inlet microbe flow, and this is the wasted microbe flow. So inlet minus wasted will tell you how much of microbe flow is present inside the tank, and XE is your effluent concentration or the microbes in the effluent. And XR is the microbes in the wasted sludge. So this first expression is actually how much of microbes that are coming out in along with the wastewater, and the second expression tells us how much is the microbes in sludge. Yeah. So microbe flow and outlet is basically the microbe concentration which are coming out of the tank and along with the treated wastewater which is going to enter into the next process. And this expression is basically the amount of microbes which are going to be disposed as sludge. So both of these together will tell us how much microbes are going to exit the tank at any point. And then we have the next term, which is, now this is the rate of growth of microbes in the tank. So that is represented by Rg. So, and V is the volume of tank. So this Rg slash V, that is given by the formula minus Y into C minus Kd into X. Now this y is your yield coefficient. Yield coefficient is nothing but the mass of VSS, or VSS here is bacterial or microbial concentration or mass of bacteria. So VSS or volatile suspended solids are bacteria because bacteria are also volatile. So they also come under the category of volatile suspended solids. So mass of VSS divided by the mass of BOD removed. Now why are these two forming a ratio here? Because the amount of BOD removed or amount of organic matter removed will be directly proportional to how much of bacteria has grown after consuming that organic matter. So on the one hand, you have BOD which is reducing, or the organic matter that is reducing. So so this will correlate with bacterial growth. Microbial growth. So when you have BOD which is consumed by the bacteria, so BOD will reduce and there will be a growth or increase in the mass of bacteria and mass of microbes, and these microbes are also represented by the volatile suspended solids. So since they are volatile in nature, so that is why we also denote it as VSS or volatile suspended solids. Now yield coefficient tells us how much of microbial growth is there per mass of BOD removed. So that is your yield of bacteria or the growth of the bacteria. How much of BOD is consumed to yield how much of bacteria. So that is what this ratio tells us. And then there is RSU, which is ratio of mass of cell formed to substrate utilized. So substrate is nothing but organic matter. And then X, which is coming here. So this, I mean, Y and RSU are the same term. So it's not like a multiplication of Y with RSU. X is the concentration of microbial mass. Okay, is it clear whatever I explained so far? So here, what we are trying to do is we are trying to find the concentration of microbes present at a given concentration inside the tank. So that's why we go by the mass balance, microbes at the inlet minus the microbes at the outlet plus whatever is present in the tank, the total sum will be equal to 0. What this means is whatever microbes are coming inside, they will multiply and grow. And if we add the inlet microbes to the microbes that have grown in the tank, that should be equal to the microbes that are exiting the tank, either as sludge or along with the treated wastewater. So this is the balance, and to find out how much of the concentration is there, we have used this equation where this is the inlet, Q is the inlet flow rate, X naught is the inlet concentration of microbes, minus the outlet concentration, outlet mass of microbes. So for finding out the outlet mass of microbes, we have to add the mass of microbes which are exiting as sludge or wasted sludge plus the mass of microbes that are exiting along with the treated wastewater. So that is what we have added here, plus the growth of microbes inside the tank. So the sum of these three terms should be equal to 0. Is this clear?
Yes.
So based on this, there is a derivation. I think you can watch the derivation in the video lectures. So I don't have to explain it again here. Okay, so in activated sludge process, we don't just have to worry about microbes. We also have to look at how much oxygen is required for their growth. So coming to oxygen requirement. So the mass of oxygen required per day equals okay. So the mass of oxygen required per day equals the mass of BOD ultimate removed minus 1.42 times the total active mass of organisms wasted per day. So we've discussed about BOD 5, but do you all know what is BODU or ultimate BOD? What is ultimate BOD? Okay, so can you at least tell me what is BOD 5? Five-day BOD. Okay, BOD 5 is the amount of oxygen required to break down organics by the bacteria at 20 degrees Celsius for 5 days. So the difference in the oxygen levels before and after the 5 days will tell us how much oxygen has been consumed. So that will tell how much of oxygen is required by the bacteria to consume that organic waste. Okay, so then what is ultimate BOD? So ultimate BOD is if we extend the time of the breakdown from 5 days to infinity. So if you just leave the sample along with the bacteria for infinite number of days, so there is a limit up to which the bacteria can oxidize the waste, right? Beyond a certain amount, it won't be able to break down. Certain organics cannot be broken down by bacteria, and that limit up to which the bacteria can break down organics is called your BOD ultimate, or the oxygen required for breaking down the maximum amount of organics by bacteria is called BOD ultimate. So, amount of oxygen required per day is your oxygen demand for ultimate BOD, or the ultimate oxygen demand, minus 1.42 times the total active mass of organisms wasted per day. Now, why are we subtracting these two terms? It's because the amount of oxygen required per day, whatever oxygen is being supplied, some of it will go for oxidizing the waste or organics by the bacteria for their growth and respiration. But some of that oxygen will also get used up for oxidizing or consumption of the dead cells. So, as I told before, during the activated sludge process, the bacteria will die, and the dead cells also need to be oxidized by the bacteria. So, they will also become organic waste, and they also need to be oxidized by the bacteria. So, whatever oxygen we are supplying, some of it is going for organic. Some of it is going for oxidizing the organic waste, and some of it is getting diverted towards oxidizing the bacterial cells. So, if we want to calculate exactly how much oxygen is being used up for only the organic waste, then we have to use the formula, this formula. So the ultimate BOD will also include whatever organisms are dead, or whatever oxygen is required for oxidizing the dead organisms. So that we have to subtract. So that is why we are using this formula. So mass of BOD you remove minus 1.42 times the total active mass of organisms wasted per day. So amount of organisms wasted per day, that we can calculate, that we can get from the sludge that is being wasted, like what that is being removed per day. So if we subtract these terms, then we will get how much of O2 is required for oxidizing only the organic waste. Any questions? No questions? Okay. So, how do we supply the oxygen? So, there are different mechanisms with which we can do aeration or supplying oxygen. So, we will look at them one by one. So, the first one is the conventional method. So, in the conventional method, we have aeration and mixing throughout the tank. So, if this is the tank, then throughout the length, we have aerators and mixers so that the air is properly, so that the oxygen concentration is maintained. The second one is tapered. So in tapered, the aeration is provided at the inlet alone. So at the inlet, there is a large number of aerators present, maybe multiple aerators throughout the width of the tank also. But towards the middle and towards the outlet, there is very less number of aerators. So this is because we can reduce the O2 consumption by this, because at the inlet, when the microbes just enter the tank, that is where they will start consuming the organics, and that is when they need maximum oxygen. So when we provide aeration at the inlet, compared to what we provide at the outlet the organisms are just they are multiplied, and they have reached the end of their life, so they don't need to survive. I mean, they've treated all, they don't have any more food for breaking down, so anyway they are going to die. So instead of providing aeration at the outlet, we are intensifying the aeration at the inlet, so that the microbes will uh, break down the organics even more fastly. And this also increases the efficiency of the process, because if we provide equal number of aerators throughout, then it won't be as it won't give that much oxygen as compared to when we give more aerators at the beginning, so that your oxygen concentration at the inlet is higher. So when the oxygen concentration is higher at the inlet, the process immediately starts, and that is why it is more efficient. The next one is step aeration. So here, in this kind of tank, there is no one inlet, there are multiple inlets. So this is the first, second and third. And there are aerators at each inlet. So this will ensure that just as the wastewater enters the tank, there will be aerators here also. So as soon as the water enters, so aeration will start, and microbes will start consuming the organics. And then we have completely mixed systems. So it is similar to step aeration, but here we also have multiple inlets and outlets. So we have inlets and outlets at multiple points. And at each point, the fresh feed along with the sludge are introduced. So this ensures that the demand and supply are uniform throughout the tank. That is, there is no lag, or there is no how to say. I mean, it ensures that the oxygen concentration throughout the tank is the same, and there is no excess oxygen at one place and lack of oxygen in another place. And the fifth is oxygen supply instead of air. Now, why is this important? Because ultimately the bacteria are going to use air oxygen, right? So, and air has only 21% oxygen. So if we want to ensure the efficiency of the process, it is better to supply oxygen directly instead of air. So this ensures high bacterial activity. and high settlability. Then we have the sludge volume index, or SVI. So the sludge volume index is the sludge volume after settling for 30 minutes. So like I've told before, when we discussed about the solids in wastewater, we also discussed about settlable solids, right? So I have told you before that we have something called an Imhoff cone, where we allow the wastewater to enter, and whatever sludge is there, it will settle down at the bottom, and then we measure the height of the settled sludge. So we use this to determine the sludge volume index also. So after 30 minutes, whatever sludge volume is there, so after 30 minutes, whatever sludge has been collected, the volume can be found by using the formula. So you have the height and you also know the diameter. So SVI equals the sludge volume after 30 minutes. Now this is in terms of ml per liter. Now whatever graduation is here, that's going to be there in ml. So per liter of wastewater entered, so whatever volume of sludge is collected, that will be in ml. So it is ml per liter divided by the mixed liquor suspended solids, or MLSS concentration. So MLSS is again the concentration of microbes. Now this is going to be in milligrams per liter. So sludge volume index is the amount of sludge collected divided by the concentration of the microbes. So for a good sludge, the SVI should be 40 to 100. What that means is your sludge volume should be low, whereas the MLSS or the microbe concentration should be high. Why is this necessary? This means the amount of dead bacteria will be less compared to the amount of live bacteria. And if your SVI or the sludge volume index is more than 200, then the sludge is poor. So this means organics have not broken down, so they can also become part of the sludge. So they are also settling down as sludge. So that will only increase the volume of sludge that is settled down, and that is something which is not desirable. So here we have to discuss one more term called sludge bulking. So sludge bulking is where a large volume of sludge is produced, and this is caused by filamentous bacteria that they don't settle down easily. So some type of bacteria which are filamentous, they will form bulk very easily, and their growth should be restricted. So if your SVI is high, it means that we need to we need to bring some changes so that the micropopulation filamentous bacteria is not in excess compared to your active bacteria, or the bacteria which is breaking down the sludge, breaking down the organics. Okay, coming to trickling filter. So till now we've seen how much we've seen about suspended growth process where the bacteria remain suspended in the tank. So now we will be seeing about attached growth process. So trickling filter is a type of attached growth process. So it is the second most commonly used process. So here we have something like this, a disc. So water or the wastewater will be sprinkled. So the wastewater will be sprinkled over the media. So this will be filled with certain packing material. So over this, when wastewater is sprinkled, what will happen is the bacteria will start growing on this media, and it will form a film. So this film or the slime layer uh, it's about 2 millimeter thick, and once this film is formed, as the thickness of the film grows, the anaerobic conditions will be developed at the bottom of the film. So if this is your packing material, and over this we have the slime layer. So at this point, at the bottom of the slime layer, anaerobic conditions will develop. And once the anaerobic conditions form, the slime layer will start decaying, and then it will get detached. So what happens when it gets detached, it will fall down, and then it will be collected at the bottom. So the slime layer is collected at the bottom. So the major advantage of this trickling filter is that it is prone to filter flies. So since it is not in an enclosed or it's not in a it's not in a tank where there is continuous flow. Here, since everything is out in the open, so that is why the flies can come and they can lay their eggs. So the eggs may hatch and then they can become flies. So that will create a huge problem for the microbes that are living in these trickling filters. Then we have aerated lagoons. So aerated lagoons are mostly aerobic at the top. So if this is a lagoon, so mostly because of gas exchange. The top layer is aerated. And it is aerobic. Whereas the bottom layer is anaerobic. So, in order to design a lagoon, we need to consider some factors. Like how much BOD removal is expected, the effluent characteristics or the characteristics of the wastewater that is coming out after treatment. So, those characteristics. Effect of temperature. Oxygen requirement. Energy required for mixing. And solid separation. Now, there are some drawbacks in the activated sludge process as well. The first one is that we need a large area. So, unlike trickling filters, where we can just have the filter beds placed vertically, so instead of that, we will be needing a large area and a tank for the suspended growth process. So, that is a major disadvantage of this process. So, we've seen that in an activated sludge process, there is inflow as well as outflow. So, most of the time, it is designed in a continuous way, that is, there is continuous inflow and continuous outflow. But we can also have a batch type reactor where, for example, one tank, in the same tank, we can have primary sedimentation and then ASP and then secondary sedimentation and then the disinfection. So all of this can be done in a sequence. So since all of these are done in a sequence and it is a batch process meaning that there is no continuous input or output. So there is only a fixed input, and then once the reaction is completed, only then there will be an output. So once the first batch is completely treated, the second batch will be made to enter the reactor, and then that will get treated, and then that will be sent out. So similarly, wastewater will be treated in batches. So that is why it is called a sequential batch reactor. So the first tank maybe can be used for, the same tank can be used for multiple processes and multiple batches of wastewater. So this sequential batch reactor or an SBR, this combines all the steps in one tank. So for an activated sludge process, we need two tanks at least. One for having the microbial reaction, so where microbes will break down the organics and consume them. And then there will also be a second reactor where we need secondary sedimentation. So that is where the sludge will be collected. So these two tanks are required. So that can be eliminated by using a sequential batch reactor, where in the same tank we can have first the reaction between microbes and organics will take place, and then the mixing will stop, and then the sludge will be allowed to settle down. So the first part is where your reaction happens, so that is where active mixing will be there. Second part, for the settling, we don't need any mixing, so the mixers will be stopped and the sludge will be allowed to settle down. So since these processes are happening in a sequence in the same tank and for the same batch of wastewater, it is called a sequential batch reactor. So it is space saving. And we can have 2-3 processes at the same time. We can also have aeration followed by we can have equalization followed by aeration or the reaction between microbes and the organics, and then sedimentation or clarification, all three processes happening in the same tank. So then we have operations in a sequential batch reactor or SBR. So the first one is the fill operation. So here the tank is filled with wastewater, so there is no process that is happening. So it is in static mode, there is no mixing or anything, so just wastewater is allowed to fill, enter the tank, and it is allowed to fill. So this is the first phase. So the tank is just filled, and then the next processes will happen. So there are different types of fill. The first one is the static fill, where there is no mixing, and then there is the mixed fill, where as the wastewater enters, we have mixers present. So that will just mix the water thoroughly. So in this condition, there is no aeration, so there is anaerobic conditions also. And then there is aerated fill. So here we have mixing as well as aeration. So this ensures that there is no anaerobic conditions and only aerobic bacteria are grown. if we want, we can create aerobic or anoxic conditions by switching off the aerators. So there can be periods of aerobic activity, and there can be periods of anaerobic or anoxic activity also. The next stage is the react stage. So once the tank is filled, the next is the reaction phase. So here, when the tank is in the react mode, the filling operation will be completely stopped. The tank is closed, so there is no inlet, and only mixing and aeration will be there. So there is mixing as well as aeration. And the next step is the settle. So react is the stage where microbes will actively decompose the organics and sludge will be produced. So then we come to settle. So here there is no mixing, and there is no inlet. Whatever reaction has happened, it would have formed sludge. So the sludge will be allowed to settle. So there is no influent entry, no mixing or aeration. So you can see that all of these processes are taking place in the same tank, one after the other. So this saves a lot of space. And the next one is the decant. So here, after settling, whatever supernatant is there. So at the bottom we have sludge. So whatever supernatant is present will be allowed to flow out of the tank, and it will be taken for the next process. And the last step is the idle step. So here the the tank is kept idle. So if there is no operations, once all the operations are complete, then the tank will be in idle mode. That is, there is nothing to treat, and that is the one of the phases of the sequential batch reactor. So it happens towards the end of the process. Once decant is also complete, sludge is also taken away, so then the tank becomes idle. So the duration of one cycle is 4 to 6 hours. And the cycle time, or TC, is your time required for filling, time for reaction, time for settling, time for decantation, plus time for idle. Now your time for reaction is again a sum of certain terms. They are the time required for aerobic condition to develop, and time required for anaerobic conditions, and the time required for anoxic conditions. So all of these are also important, because we need anoxic conditions for the nitrification, and we need anoxic conditions for denitrification, and anaerobic conditions for phosphate removal. So all of these also are necessary. So we can, once the BOD is removed, then we can maintain anoxic and anaerobic conditions so that nitrate and phosphate also get removed. So when we talk about a sequential batch reactor, we have something called a volume exchange ratio, which is the volume of effluent withdrawn in a cycle divided by the working volume of a reactor. So this is basically how much of wastewater is getting treated in a cycle. Divided by the working volume of reactor. So, and then we have the number of cycles per day, or NC. NC is number 24 divided by TC. The TC is the total time for the reaction, or the cycle time. So you have 24 hours in a day, so 24 divided by the time required for each cycle to run will tell you how many cycles can be run in a day. And then we have Q, or the flow rate. So the flow rate equals the volume of the tank, the filled volume of wastewater in a cycle multiplied by the NC. So this will tell us how much, VF is the volume that is treated in each cycle. That multiplied by the number of cycles will tell you how much of wastewater is treated in a day. So then, I've already mentioned about nitrogen removal. So I told you that for nitrogen removal we need anoxic conditions. So what happens actually in nitrogen removal? So nitrogen is present in the form of ammonia. So this ammonia nitrogen when there is aerobic conditions, or when there is oxygen present, so ammonia nitrogen will be converted to NO2, and then NO3. So whatever ammonia is there, that will get ammonia is in the form of NH3. So ammonia will get oxidized to NO2 and then NO3 in aerobic conditions. So this reaction takes place in then why do we need anaerobic conditions or anoxic conditions? I will come to that. So anoxic means no oxygen. Then how is it different from anaerobic? In anaerobic, we don't have dissolved oxygen, but there is oxygen in the form of nitrates or sulfates or phosphates. So bacteria can utilize oxygen in these in anaerobic. Whereas in anoxic conditions, even these are not present. Like there is completely no oxygen present. So under anoxic conditions, where we have no oxygen, so the nitrate or when there is no oxygen, what happens? The reduction happens, and whatever nitrates are there, they will be converted to nitrate, and then nitrous, and then N2O, and then finally nitrogen gas. So this is why we need anoxic conditions, so that nitrates can get converted to nitrogen gas, and before that we need aerobic conditions so that ammonia will get converted to NO3. So there are some advantages of sequential batch reactor. It's that the area requirement is less. And the initial cost is also less. And power requirement is also less. And like I said, in the same tank, we can maintain anaerobic and anoxic conditions. For anaerobic conditions, we have to give aeration. For aerobic conditions, we have to give aeration. And for anoxic and anaerobic conditions, we have to cut down the supply of oxygen completely. Someone has asked reduction. I didn't understand the question. Is there a question? Okay. So SPR also has some disadvantages, like we need proper maintenance of the tank. So even a slight, if there is a slight mechanical dysfunctioning or something, then the entire process can be stopped. And also it is a highly sophisticated process where we need to maintain exact times for all of the reactions, and exact conditions need to be maintained. So in the same tank, since we are going for aerobic as well as anaerobic and anoxic, we need to be very careful with maintaining the exact conditions, and that requires a high level of sophistication. Any doubts or questions?
No, ma'am. Okay, so next we'll see about membrane bioreactors in brief. So membrane bioreactor is where we have a membrane. So membrane is more or less like a sheet, and on this we can have bacterial growth. So we can just suspend this sheet, and then we can suspend this membrane, and the membrane will, of course, it will have pores through which wastewater can flow in and out, and because of the flow of wastewater, bacteria will grow on the surface of the membrane, so that once the bacteria has grown completely, when there is an inflow of wastewater through the membrane,
So the wastewater will get treated as it passes through the membrane because bacteria is present, so that will degrade the wastewater as it passes through. So that is the advantage of a membrane bioreactor. And again, it is a type of suspended growth because it is going to be placed in a tank. So there we also have suspended bacteria present. Okay, then coming to phytoremediation. So phytoremediation is using living plants to remove pollutants. So here organics are broken down, and whatever organics are broken down, they are uptaken by the plant itself, and metals also get uptaken by the plant or the roots. And once the treatment is complete, the plant is cut and it is harvested. So once it is harvested, it is properly disposed. So it is suitable for on-site removal. So for this, since the plant is uptaking the pollutant after breaking it down, we have the bioconcentration factor defined. So the bioconcentration factor, or BCF, as we had discussed previously, it is the concentration of the pollutant present in the living organisms divided by the concentration present outside. Like for example, if we are discussing about a fish, then the amount of pollutant present per kg of the fish divided by the pollutant concentration outside in the sea. So that is your bioconcentration factor. So we are going to define a similar term here. So for phytoremediation, the bioconcentration factor is defined as the ion concentration in the plant divided by the initial ion concentration in solution. And then we can also define another term called the translocation factor. So the translocation factor is the ion concentration in stems or leaves So this is in micrograms per gram divided by the ion concentration in roots. So this is because it is assumed that if we have phytoremediation happening, then the pollutant travels from the root, from the soil to the leaves and the shoots. So that is why we are defining this term. So this tells us how much the pollutant has traveled in the plant's body. So if the ion concentration in the stems or leaves is more, then compared to the roots, then it means that the plant has successfully uptaken and it has transferred the pollutant to its shoots. Advantages of phytoremediation process is that it is low cost, it is also energy efficient, and it preserves the natural state of environment. But on the other hand, it is useful only for low or medium level contamination. And we also need a longer time for treatment because phytoremediation is a slow process. So the plant has to grow and then assimilate the pollutant. So it takes time. And also, bioconcentration is possible because of which the plant needs to be properly disposed. And we can also have microalgae based treatment. So, macro or microalgae based biotransformation of pollutants. So for this, we can grow algae in photobioreactors, which is basically a reactor where algae is allowed to grow under the presence of a light. So then we can allow the wastewater to come in contact with the algae so that the algae can break down the organic waste. And then it can be separated by filtration and flocculation. Okay, so we've come to the end of today's lecture. So we'll look at some questions and answers. The first question is, what is the efficiency of extended aeration process? So an extended aeration process is where we provide longer duration of aeration. So this is mainly seen in an aeration pond. So we give a longer aeration time so that the organics are broken down even more efficiently. So the options are 60 to 80%, 90 to 95%, so the correct option is 90 to 95%. So because the aeration period is high, the organic loading is low, so that is why the process is highly efficient. So the question is, which of the following can be done to prevent sludge bulking? So the options are, first one is to provide sufficient aeration. Second one is to increase the retention time. The third one is by reducing the flow rate. And the last one is none of the above. So the first option is correct. It's because when we provide sufficient aeration, it prevents the growth of filamentous bacteria. So once the filamentous bacteria is controlled, then the sludge bulking will be reduced. The question is, phosphorus removal occurs in which of the following conditions? The first option is aerobic, second one is anaerobic, the third is anoxic, and the last one is facultative. So in aerobic conditions, in the presence of oxygen, phosphorus is not going to be removed. It is only removed by anaerobic bacteria in the absence of oxygen. And in anoxic conditions, we've seen that nitrogen removal and denitrification takes place. And under facultative conditions also, phosphorus is not removed. So the question is, what is the ideal COD to nitrogen ratio for sludge granulation? So sludge granulation happens when we allow the particles of certain particles, when they are present in the along with microbes, so the microbes will develop a layer. So this slime layer is formed over the particle. And once the slime layer is formed, so it's something like this. The top of the slime layer will be aerobic. So aerobic conditions are useful for removing BOD and nitrification. At the bottom of the slime layer, there is also anoxic conditions developing here. So this anoxic will be useful for denitrification. And at the bottom we have the anaerobic condition, where phosphorus is removed. So this is granulated sludge, and this is very useful because here all the processes are taking place in a concentrated manner so that it's not spread out, and so area requirement is also going to be lesser. So the biomass is also highly dense and it is tightly packed,
and this ensures that the process is also more efficient. So that is sludge granulation. So the question is, what is the ideal COD to nitrogen ratio for sludge granulation? So the correct answer is 2 to 5. So this ensures that there is less growth of heterotrophic bacteria. The question is, which of the following plants can be used for removing chromium and lead? So the options are mustard, so the options are Indian mustard, willow, smooth water hyssop, and sunflower. So the correct answer is Indian mustard. So it is very effective at removing chromium and lead. Okay, so with this, we've come to the end of so with this we've come to the end of our lecture. So if you have any doubts or questions, let me know, and